Hi everyone, these are the lecture notes for Chapter 5, Structure and Function of Plasma Membranes. The functions of the plasma membrane are defining the outer border of all cells and organelles, managing what enters and exits the cell, receiving external signals and initiating cellular responses, and adhering to neighboring cells. And before I move on, I know I just read the slide to you and it sounds kind of boring, but um, before I move on to discussing the plasma membrane structure and function in more detail, I just want to say this is probably one of the most important chapters in biology just for everyone in general, no matter what you're going to do with your life or um, what career you end up in, just for people in general, because so many diseases really are the problem is with some component of the plasma membrane. I'll give you an example, diabetes. So a person who has type 1 diabetes, there is a protein on the plasma membrane of your liver cells. It's a receptor. So insulin binds to that receptor and it causes those cells to take glucose from your blood and lower your blood glucose levels or your blood sugar. That's what a lot of people call it. Um, so there's a problem there, and whenever you don't have insulin, when a type 1 diabetic does not produce enough insulin, but if you have type 2 diabetes, um, you may produce plenty of insulin, but it's not binding with the receptor properly, so it's still not doing its job. But the point is, there's so many diseases and disorders that come from, and health problems that come from, originate with plasma membrane structure problems and function problems. So let's talk about the plasma membrane structure first. The illustration that you see here, that you're going to see many times throughout this lecture, is called the fluid mosaic model, and that is the name given to the model for the plasma membrane. Um, there's the phospholipid bilayer, the double layer of phospholipids. Remember, phospholipids have a hydrophilic phosphate head and hydrophobic fatty acid tails. And those fatty acid tails point toward each other because they are nonpolar and insoluble in water. And then the phosphate heads are hydrophilic, so they point to the outside and inside of the cell where they come in contact with water. So you see all of these phospholipids, phospholipid bilayer. And then you see cholesterol. Cholesterol can be found embedded within the phospholipids. And then you see these blue structures, these large blue molecules. They're proteins, and these proteins have different functions. Sometimes they have an opening, so they act as a channel. They allow passage of ions and small molecules from the inside of the cell to the outside, or from the outside to the inside, that they allow passage across the plasma membrane. And you also see these carbohydrate chains, and they can either be attached to a lipid, then if they are, they'll be called a glycolipid, or the carbohydrate chain can be attached to a protein, and it's called a glycoprotein. But many of these glycolipids and glycoproteins function in cell recognition, identifying the cell to your immune system. And it is what doctors use to determine if someone is compatible to donate an organ, if someone needs a transplant, if their tissue compatible is compatible with the recipient. So the main component of the plasma membrane is the phospholipid bilayer. So this is an image of a phospholipid in detail. Phospholipids are molecules that are called amphiphilic, and that means that it's got a hydrophobic region and a hydrophilic region. The hydrophobic region, hydrophobic means fear of water, but the hydrophobic region is the region that's insoluble in water. It's nonpolar. So that is the two fatty acid chains. And then the hydrophilic region is a phosphate group, and it is polar, so it is soluble in water. So the phospholipids arrange themselves in a double layer to where the fatty acid tails face each other and the heads face outside and inside the cell. Now, the fatty acid tails can be saturated or unsaturated. If they're unsaturated, they have at least one double bond, remember, and they'll have the unsaturated fatty acids will have a kink, a little bend in their chain. This is an illustration simply of a phospholipid bilayer without the proteins embedded. The fluid mosaic model is called fluid mosaic because the molecules are actually fluid. They actually move in and around and past each other. They don't jump around, but they sort of move, just kind of move among each other slowly. So they're fluid in the fact that they move. They're not just stuck in one spot. And the mosaic is because the proteins and other molecules are scattered throughout the lipid bilayer in a mosaic fashion. So this is just showing a lipid bilayer without the proteins and the cholesterol molecules, and it's showing you that the polar heads are facing outward and the hydrophobic tails are facing inward. If you were to just drop a sample of phospholipids into a beaker of water, they would automatically form a structure like this. I mean, they would do that naturally. They would automatically form a bilayer. And that is because those hydrophobic tails are going to orient themselves in such a way that they're not in contact with water. And then the heads are going to orient themselves in such a way that they are. So they form that bilayer naturally. Now, we talked about the phospholipids. The next thing is the proteins. The proteins are the blue molecules that are scattered throughout the phospholipids. They have various functions. We'll talk about the different types of proteins, but for now, they can transport molecules like the channel proteins. They can transport molecules across the plasma membrane, either into or out of the cell. They can function as receptors, like the example I gave you for insulin. Insulin would bind to a receptor protein, and then it would cause the cell to lower your blood sugar levels. They can function as enzymes. They're proteins, so if their role is to speed up a chemical reaction, they can function as enzymes even though they're attached to the plasma membrane. They can also function in binding, either binding the plasma membrane to a structure inside the cell or binding the plasma membrane to another cell, and adhesion, which is similar, causing the cell to, many cells group themselves into tissues, and so they're going to have to be joined in some way. So some of the proteins are used to join one cell to another cell. Integral proteins are proteins that go all the way go all the way through the entire phospholipid bilayer. They have one or more regions that are hydrophobic and then other regions that are hydrophilic. And the location and number of those regions determine how they arrange themselves within the bilayer. But if they're integral, that means they go all the way through the lipid bilayer. A lot of what determines how a protein arranges itself in that phospholipid bilayer are these characteristics in this chart. So if the amino acids on the outside of the protein are nonpolar, then they're going to tend to be found within the fatty acid tail, within the hydrophobic region of the bilayer. If the amino acid is polar, then that means it's going to be soluble in water, or if it has a charge, whether it's positive or negative. Either way, polar or a charge, then it's probably going to be located either on the outside, attached to the phosphate heads on the outside of the cell, or on the inside, attached to the phosphate hydrophilic heads. And then here's another group that are nonpolar. So the nonpolar will tend to be located within the fatty acid tails. The carbohydrates, there are two examples of where you find carbohydrates. They're always on the outside of the plasma membrane, the exterior surface. And if they're bound to a protein, they form a glycoprotein. So the carbohydrate portion is the glyco part. And if they're bound to a phospholipid, they are called glycolipids. And they function in cell recognition, and some of them also function in attachment. Because they are carbohydrates, if you think about it, one carbohydrate is sugar. So carbohydrates tend to be sticky. They tend to be adhesive. And so some of those glycoproteins and glycolipids also function in making the outside surface of the cell kind of sticky, so that it can adhere to another cell or tissue or substance. All right, so we'll start with the types of proteins that are found within the phospholipid bilayer. One is called a receptor protein. Now here is a different example. I gave you the example of a receptor protein for insulin, but there's a receptor protein for all kinds of different chemicals. Our immune system's T cells. Now the T cells in our immune system help to destroy specific viruses or bacteria or pathogens. So our immune system has these T cells that have receptors called CD4 receptors. And they are glycoproteins, and they recognize the HIV, which is human immunodeficiency virus. They recognize that virus as self. And that is how HIV is able to trick our immune system into infecting our cells and tricking our cells into making more copies. So that's just one example of a receptor protein that has a function that we don't like, but sometimes those receptor proteins can be manipulated by viruses and other pathogens. But what it also does, by the way, I forgot to say this, but what it also does is when that T cell binds to that virus, HIV is different because it ends up destroying the T cell. But sometimes T cells have receptor proteins for viruses that just cause it to recognize the virus. And then the T cell will begin to make more T cells that can recognize that virus, many, many millions that can recognize that virus, and they will go around and destroy the virus. It doesn't happen with HIV because HIV actually destroys the T cells. That's why it's so dangerous, because it pretty much shuts down our immune system. The fluidity of the membrane is affected by different things. It needs to be fluid, but it doesn't need to be as fluid as water. It needs to be kind of have a little bit of sturdiness to it. So it's got to be flexible, but also be able to maintain its structure. So the phospholipids, when they have more saturated fatty acids, the membrane is more rigid. And when they have more unsaturated fatty acids, the membrane is more fluid. Cold temperatures cause the membrane to be more rigid, and warm temperatures cause it to be more fluid. And cholesterol within the membrane keeps the membranes fluid when it's cold, and they keep it from not being too fluid when it's hot.